What comes to your mind when I say your best friend? Your sibling, someone you grew up with, someone so permanent that you couldn't imagine life changing them at all. For me, it was my grandmother. I have spent hours with her talking, learning. She would tell me stories about the universe, that everything follows a rhythm. Things are created, maintained for a while to serve its purpose, and then destroyed. For me, these were just stories. And then I visited her, and suddenly she was slower, smaller, sometimes more fragile, and sometimes she couldn't recognize me. But yet her embrace felt exactly the same. That made me ask a question. Why do we change so profoundly with age? It went back to the same story she used to tell me about the universe. In our cellular system, the same rhythm is followed. Things are created. These are fundamental functional unit of life, proteins, constantly being created, maintained to serve a function, and then destroyed. This balance of cycle is called proteostasis. When we're younger, this cycle is maintained efficiently. However, when we grow old, there is an evidence of damaged macromolecules that needs to be destroyed and degraded, keeps accumulating. And it points to a malfunction in a machinery that is supposed to maintain this cycle. And one of these key systems is called proteasome system that shows remarkable decline in its function with age. And it causes proteostasis imbalance. And this imbalance manifests into neurodegenerative disorders and even metabolic syndromes. The proteasome system is actually a giant protein machinery. It is made up of different components, such as the regulators that manages its function, the core catalytic component that actually does the activity, and it utilizes energy and signaling molecules such as ubiquitin. The damaged molecule enters this protein and enzymatically cleaved into smaller pieces. These activities could be of different types: chymotrypsin-like activity, caspase-like activity, or a different type of cleavage like trypsin. These make a standard proteasome. However, under stress conditions such as oxidative or inflammation, we see a special type of activity that resembles the standard proteasome activity. However, these are performed by special subunits. And it makes immunoproteasome, which are key in diseased condition. There are also different types of subpopulation found of proteasomes, such as the giant proteasome called 30S that has two regulators attached to it, one with only one regulator called 26S, both utilizing energy and signaling molecules. However, just the catalytic component can also perform degradation of special substrates, and these are called 20S. So I embarked on this journey to find the role of proteasome in aging and how it changes through age. For this, I would like to study the different types of activity and how it changes through the lifespan. For this, I utilized native electrophoresis method that requires fluorescent tags that helps distinguish the activities between the three subpopulation. To find the abundance using simple Western blot analysis, across different tissues of mice, such as liver, kidney, brain, heart, and lung, and also use mass spectrometry analysis to see the changes in its structure throughout age. So first of all, if I want to see what's the association between how much abundance of these proteasome particles are present in cell and how much activity it performs throughout age, in the two ATP utilizing populations, we see a trend. First of all, in liver, I saw that the abundance of proteasome in liver did not decline with age. Similarly, its activity was also maintained. However, it wasn't true for kidneys. Its abundance declined throughout age, along with its activity, completely opposite to how we predicted in liver. For other tissues, it wasn't as simplified. Although their abundance declined significantly throughout age, their activity, such as in heart, did not decline. It was maintained throughout age. Similarly, in lung, both the population had similar maintained activities with declining abundance. Also in brain. However, the exception still remains with liver, where none of the abundance or activity declined throughout the age. This challenges the traditional view of aging, where it thinks proteasome, the number of proteasome would be associated with the amount of degradation that it follows. However, with different tissues, it's not the case. We see sometimes decoupling between the abundance of the proteasome population to its activity. This reframes aging as a question of not a mere deterioration, but a remodeling process. Remember the core catalytic component 20S? We found that the association between abundance and activity in the immunoproteasome had a very distinct pattern. However, especially in brain, we could not find any activity of proteasome in the immunosubunits. That really shows that not all tissues age the same, especially in brain that lacks immunoactivity at all. Could be the reason why is it susceptible to age-related diseases. We also used mass spectrometry analysis to see the compositional subunits in different tissues such as brain, but also in different age groups. These lines that you see show us different subunits of the entire proteasome. And in brain, we could confirm that there were no immunosubunits. That's why we did not see its activity. We also saw a special modification through these analyses that was not found in other tissues. For example, in liver, there were two types of the core catalytic component subunits present, but not in brain. These analyses show us that at every developmental stage, proteasome resembles in different states. And it's not enough to ask what is the abundance and what is the activity performing these key cellular function, but it's also important to consider which catalytic state is performing these activities. And there are various states available. Lastly, we wanted to see how the structure is stable or not if we provide it with collision energy. And we see that the complex, when provided with a lot of energy, can break down in its compositional subunits. We use this method to test the stability of the complex between the infant and the old. And we see that these complexes, after giving energy, completely break down in case of infant. However, they still remain stable and intact in case of old. This shows us that although infant complexes that perform really well are really fragile and they can be really unstable compared to the adult proteasome. This may show us that although when we grow old, our proteasomes become rigid, but it might not be a constructive modification. It might bring us some destructive properties, which could affect its activity in case of stress, such as oxidative stress. So to summarize, we saw that different activities of proteasome reshapes the proteasome activity rather than simply shutting them down. We saw that even though there are smaller particles present of proteasome, it might perform a lot more activity throughout the age. We also saw that different tissues have different properties of proteasome, and it's not efficient to generalize. And we see that with age, the proteasome architecture itself changes. So to conclude, my work shows that proteasome adopt distinct molecular identities throughout the age, and it may involve complex structure and functional remodeling with age. Thank you so much.